Okay, so in this, we're going to talk about some chemical reactions that usually what occur to histones. There are some chemical reactions that are very important to histones. All right. So what we are going on is we'll talk about histone methylation. We'll talk about histone acetylation. We'll talk about histone phosphorylation. Alright, so for what we have for this, of course, methylation should be the process of adding a methyl group. Acetylation should be the process of adding in an acetyl group, while phosphorylation, you are basically adding a phosphate to it. So first of all, you should just know that there are actually three major types of what post-translational modification. That's after the histone is made. These ones, they come in to like reshape everything, rearrange everything. All right, methylation, acetylation, phosphorylation, just like that. So they occur on histone proteins. So these chemical changes, they regulate how tightly the DNA will be packed. All right, and thereby they influence the type of genes that will be expressed. They influence the type of genes that will be repressed. All right, they help the DNA to repair itself, and they make the cell cycle to continue. So what is a histone methylation? It's just addition of methyl groups. All right, addition of methyl groups. All right, addition of a methyl group. Okay, so enzymes needed include histone methyltransferases. All right, then histone demethylases. That one is to remove the methyl group. All right, so functional impact is just basically to activate or repress transcription. This is depend on the site where the methyl group is added. All right, and where the methyl group will be added depends on what result is needed. Then histone acetylation, so just like what we can see here, addition of an acetyl group, all right, to lysine residues, it neutralizes their positive charge. Enzymes include histone acetyltransferases. Then sometimes if the acetyl group needs to be removed, is histone deacetylase. All right, so function is basically to loosen the chromatin structure, increase DNA accessibility. So if you want something to be able to access the gene, okay, you add an acetyl group so that it will open it up, it will loosen it up for that thing to actually access it. So biological roles is regulate gene expression, cell cycle, and also response to environmental signals. Then for the phosphorylation, which is this stage, you just have to do it addition of phosphate group, okay, to the serine, thiourine, or tyrosine residues. So enzyme involved is a protein kinases. All right, if it needs to be removed, is the protein phosphatase that will be involved. So functional impact is just to alter the chromatin structure, recruit DNA repair proteins. So it's often linked to DNA damage response, mitosis, or if the cell wants to naturally die, that is apoptosis. So biological role is just for chromosome condensation, transcriptional regulation, genome stability. Okay, so that's it for these three chemical processes that usually occur in histones.